genetics at Purdue University. He's an entomologist specializing in honeybees, and he's come through degrees in biology and plant pathology. He's interested in the ways that bees defend themselves and was part of the USDA-funded multi-university inquiry about CCD. His observations on the grooming behavior of Varroa's original Asian bee host has led him to investigate similar behavior in European bees, grooming behavior. He's both a scientist, a bench scientist, and a field researcher who is close to beekeepers. He's a founder and trustee of the Heartland Apicultural Society, which spans several states in the Midwest, and he's organized testing for this grooming trait. It's interesting that his is the only project in the United States in a university setting where there is breeding that is being done for particular traits. His talk, Breeding Bees for Behavior Resistant to Varroa. Dr. Greg Hunt.
Hi. Whoa, that's pretty loud. How's that work? Can you ever, is it too loud or is it good? This is the best place to have a beekeeper meeting that I've ever been in. And I want to thank you all for having me and Mia for putting me up and letting me pet her Airedale, because I have a couple Airedales at home. I'm going to talk about our breeding program, and I'm interruptible if there's any questions at any time. I didn't bring a slide advancer, so I'm going to stand here and advance these. I'm originally from Ohio. I got my degree at UC Davis, so I'm familiar with the valley here. Now I'm back in the Midwest, and we have winter there. And this is one of our apiaries. You all know that we're having problems with bee die-offs, and so this is the losses from the Bee Informed website, which is the national survey where they tell how many bees, they survey beekeepers, and you can see the losses here in Illinois, 62%, Indiana 49%. This is the year before last, and it's pretty well agreed upon by bee researchers that the main cause of colony death worldwide is Varroa mites. Of all the things we can measure, pesticides are definitely a problem. But of all the things we measure, Varroa mites are the biggest problem. Mainly because of the viruses they transmit. So if we could do something about the Varroa mites, we could make a lot of we could have more sustainable beekeeping. We have products that work to control Varroa mites. These are all the products that the EPA lists that are now available, registered for Varroa mites. You know, when I moved to Purdue 20 years ago, we just had Apistan, and the mites became resistant. Now we have all these things. And the ones with red bars are basically soft chemicals. They're really not pesticides. And they work to control mites, except maybe the sucroside. I don't really think that's very effective. But the problem is we don't want to use them. We honeybee extension people tell beekeepers, well, you should treat to control the mites. But a lot of people don't want to, or just don't, and so we have to come up with a better solution. And I think that the solution is breeding And I realized slowly that the best resistance to varroa mites are behavioral solutions, which is good because that's what I'm interested in. And you know, we're all breeding for bees that survive the winter, you know, because we're all breeding for survivors. We talk about survivors, bees that survive without treatment. And we're all breeding for survivors because we never raise queens from the dead hives in the spring. And so we have winter, and in the north central region of the U.S., we're especially concerned about bees that are winter hardy. So this would be, I think, the easiest way to select for varroa resistance. This is how we used to do it at Purdue from 1997 to 2007. We would measure our varroa levels, and I think everybody should monitor their mite levels now and then. And an easy way to do it is with sticky boards. So you put in you have a screen over this sticky board, and mites are always dropping, and that's, if you see how many mites drop in a day or 2 days, that gives you an idea of how many mites are in your hive. So we bred from bees that did not seem to grow mites. We would test their mite levels 2 or 3 times a year, and if those colonies that weren't growing mites, those were the ones we raised queens from. And if you're, so I'm talking about, you know, any of us could breed for resistance to mites if you have time on your hands and enough hives. But you need to keep good records. You need to have marked queens so that you know that the bees in that colony that you're taking the data from are her daughters, and not a supersedure queen. So keep good records. So basically, bee researchers have found 2 traits that seem to be the most important for resistance to mites. One of them is varroa-sensitive hygiene, and this is the detection and removal of infested brood. And I think what's really going on here is probably they're detecting brood that has virus. But when they originally started, when they found this trait, the way they did it was John Harbaugh on the left there and Jeff Harris of the USDA Baton Rouge Bee Lab, they got a lot of queens from beekeepers all over the country, and they put them into nukes that all had the same starting varroa mite levels. And the way they did this was they made these mega packages. They shook a bunch, in the upper left there, they shook, I don't know, like maybe 100 pounds of bees into that package, and then they just prayed that they wouldn't overheat. They had a tube going down the middle and ventilation, and they quickly aliquoted the bees, same number of bees into each nuke, and put a different queen into each nuke. They found out which colonies were growing lots of mites and which were not growing mites. And then eventually, after a couple of years, they realized that the mechanism was this varroa-sensitive hygiene. The resistant bees were uncapping and removing brood. And so like that's the varroa mites reproduce in the sealed brood, and on the left there, that shows a mite that's reproducing, but the bees uncapped it and started to chew that pupa or remove that pupa. And the one on the right, that mite never reproduced. And so, since it didn't reproduce, it didn't really feed on the pupa, it didn't make the pupa full of virus, and they didn't uncap it and remove. So when they first found this, they thought that somehow the bees were suppressing mite reproduction. But what was really happening, and so they called them SMR bees, suppression of mite reproduction. But what was really happening was that the bees were removing the infested or mite fed upon pupae. And if you go to extension.org, Bee Health, it tells you how to select for varroa-sensitive hygiene. There's basically two methods. There's the easy method that's faster and less exact, and then there's the hard method, which is more precise. The easy method is to put mite-infested brood into your colony or look for signs of the behavior, which is uncapping of apparently healthy pupae, and maybe chewed cappings. And sometimes the bees recap. Like one bee will come along and say, this doesn't smell right. She uncaps it, but she doesn't remove the pupa. Another bee comes along and says, this should be capped because it's time to cap, and she recaps. The other method actually involves uncapping cells and looking for reproduction of the varroa mites. And this would be done at stages like those bottom two stages where they're starting to get dark eyes and the pupa are starting to change color. If you uncap it at that time and you find an adult female varroa mite and she hasn't reproduced, then you know she hasn't reproduced. And there's also another website, the European website, the Colos site, which also shows how to measure for this trait. And they have kind of a more detailed picture of the life cycle of these mites. Kind of interesting how they go through these different stages. You get the question.
So, if you could go back a slide, if you uncap, 2 slides, I'm sorry. If you uncap a cell and you find a female varroa mite in there, wouldn't you conclude that it did reproduce if there was still viable pupa in the cell?
No, because, well, it would be you'd feed the female it would be the female that invaded that cell. So if you only find one adult female, that's the one that invaded and failed to reproduce.
But why would it fail to reproduce in a viable cell?
Well, that's a good question. For some reason, I've been told like 10 to 15% of mites just are not reproducing. Maybe she didn't get mated. They have to mate inside the cell before the bee emerges. They mate with their brother because the varroa mites are haplodiploid like honeybees. First she lays a male unfertilized egg, and then she lays female eggs. And so they mate with their brother. And so if you just find one female adult, she hasn't reproduced.
Okay. But how is that, how do the bees control that behavior?
The bees don't control that behavior. What they do is they detect something that doesn't smell right because the reproducing mites feed on the pupae and they give them virus.
Right.
And the pupae gets sick, and the bees smell that and they uncap and remove.
Right.
And so what you're left with are the reproducing ones are the ones that they uncap and remove. So what you're left with are those 10% that weren't going to reproduce anyways.
Okay.
So and this just is back at the other website. You can look at that, and you basically put them into 6 categories: mother dead, mother alive, no offspring, which means no reproduction; mother plus only one male, no reproduction. Mother plus, if there's the mother plus another female mite and a male, they reproduced. But you know, if the mite's doing good, there should be 3 or 4 daughter mites coming out of there.
Do we know how to tell the females from the males in mites?
Yes, because the males are smaller and more circular, and they never really color up. They stay kind of pale, and they don't survive outside of the cell. An easier way is, and this comes from the Baton Rouge Bee Lab, is that you take a comb that has mites in it, and you put it in a hive, and so this is the one comb cut in half. The one on the left was putting it in a hive that wasn't showing VSH trait. The one on the right, they've uncapped and in some cases removed pupae. So that's one way, an indication that they have varroa-sensitive hygiene behavior, VSH. And we've seen this in our bees because back when we were breeding just for colonies that don't grow mites, we did introduce one VSH colony from Baton Rouge and one Russian colony and lots of survivor colonies. And I took this, this is from 2014, one of the colonies that we grafted from. They had good grooming behavior, but they were also really suppressing mite population growth. So I took this drone comb, put it in the colony for 48 hours, and then it looked like this. So if I go back and forth, you can see they removed a lot of pupae, and those white ones have been uncapped but not removed. So that's VSH, but now what we're focusing on is grooming behavior, because there's